Good morning. Today we are going to study new topic in microbiology that is some about some infectious subviral particles. We have already studied the structure of viruses and the multiplication cycle of viruses and uh, about the viruses infecting bacteria, that is bacteriophages, etc. And in your syllabus, hope you have read the syllabus and in your syllabus only very brief study about the subviral particles, that is prions, viroids and virusoids is mentioned. So first we will pass on to the first subviral particle, that is prions. Prions, uh, first of all, prion, from the word itself, prion, it is a protein. The word protein, it starts with PR. The first two letters of protein is PR, and the first two letters of prion is also PR. So in that way you can reconnect it. Okay. So only difference is that protein is beneficial to us, but prion is infectious to us, or it is harmful to us. Not only to human beings but also to animals, plants, etc. Okay, so prions, it is a type of protein that can trigger normal proteins in the brain to fold abnormally. So what happens is that it is normally found in the brain. So what happens is that it causes the normal protein to fold abnormally. Since it occurs in the brain, it normally affects the neural tissues and causes certain neurological disorders. So it is we can define prions as an infectious subviral protein particles devoid of nucleic acid. There is no nucleic acid in prions. And also the prions are very smaller, or about 100 times much smaller than the viruses. And the scientist who discovered prions is known as Prusner, or Stanley Prusner, and he discovered it in 1982. And he was studying a disease known as scrappy. That is, scrappy is a degenerative disease found in sheep. And while studying about the causes of the disease scrappy, he found that the disease was due to a proteinaceous infectious particle known as prion. That is how the infectious proteins known as prions were discovered. And at first, during the earlier times and all, the discovery of Prusner was not widely accepted. That is, many people told that the protein cannot be infectious, or it cannot be devoid of nucleic acids and all like that. So many the prions, so they are the infectious proteinaceous particles. And for the discovery of prions, the Prusner, scientist Prusner, he got Nobel Prize for the discovery of prions, and the Nobel Prize was in the field in of physiology and medicine, and he got the Nobel Prize in the year 1997. So that is about a general introduction about the prions. Then we can go into detail how the normal protein becomes infectious, or how it folds abnormally. And the normal prion protein is known as PRPC. Okay, that is PR, capital letter P, and R is small letter, PRPC. And it's an infectious, or the protein occurring due to the abnormal folding, it is known as PRPSC. SC for scrappy. Okay, SC for scrappy disease. So PRPSC, normal prion protein is PRPC, and the abnormal infective variant produced by mutation is known as PRPSC. So, prion is a misfolded rogue form of normal protein found in the cell. What do you mean? What is the word meaning of rogue? Rogue means it is an infectious form. That's all. Infectious form of protein. And this rogue prion protein, which may be caused by a genetic mutation or occurs spontaneously, can be infectious, stimulating other endogenous normal proteins to become misfolded, forming plaques. You might have studied about our normal neurodegenerative disease, that is Alzheimer's. How Alzheimer's is caused? It is due to the formation of some plaque like particles in our brain. In the same way, when the normal protein become infectious, it causes the formation of certain types of plaques in our brain or the neural tissue, and thus causes certain neurological disorders, or means it what to say, it disforms the brain function like that. So remember, the normal prion protein is PRPC, and when it become abnormal, it is known as PRPSC. And in this figure, you can see this is a normal protein, PRPC, represented by green dots. Okay, this is the normal protein, PRPC, and when the endogenous normal prion protein, that is PRPC, is converted into disease-causing form, PRPSC, when it encounters this variant form of protein, and it gets converted due to mutation or some other disorders. PRPSC may arise spontaneously in the brain tissue, especially if a mutant form of protein is present, or it may originate from misfolded prions consumed in food that eventually find their way into brain tissue. So, the normal protein gets converted into the abnormal protein by two ways, either by a mutation or due to the improper folding or misfolding of the normal protein. And that is when we consume, in the case of, for example, in the case of human beings, when we consume a diseased animal, or a diseased food, animal or plant food, this protein enters into our body and it affects the brain. So that are the ways the normal prion protein gets converted into the infectious or the harmful PRPSC. Okay, so in this cycle, you can see the conversion of PRPC to PRPSC. And next we can study the different diseases caused by the improper folding of prions or the infectious subviral particles prions. What are the diseases caused by the subviral particle prions? And the first, the diseases caused by prions are commonly known by the name transmissible spongiform encephalopathy. So the prions are known to cause various forms of transmissible spongiform encephalopathy in humans and animals. Uh, there are numerous neurodegenerative diseases caused by prions, and all these diseases are known by a common name, that is transmissible spongiform encephalopathy. And not only in human beings, this infectious prions causes diseases in animals and also in plants. Then uh, this transmissible spongiform encephalopathy, it is a rare degenerative disorder that affects the brain and nervous system. So this type of disorders mainly affect the brain and nervous system. What are the two organs affected due to the infectious prion infection? One is brain and the second one is our nervous tissue. And the accumulation of rock proteins causes the brain tissue to become sponge-like, killing the brain cells and forming holes in the tissues, leading to brain damage, loss of motor coordination, and dementia. What happens when the normal brain tissue turn into spongy form? It becomes functionless, or its function gets completely altered, and some holes are also formed in the brain cells. So the normal brain functioning gets altered and it causes neurodegenerative diseases. Infected individuals are mentally impaired, mainly mental impairment, and becomes unable to move or speak. There is no cure, and the diseases progresses rapidly and eventually leading to death within a few months or years. And till now, no exact medicine has been discovered for this disease. So, when a human being gets infected by this disease, he become mentally impaired and become unable to move or speak. That are the normal causes of this disease. And also the person will die within few years or within very few months. He will die. And what are the diseases in humans? Diseases in humans include Kuru, then fatal familiar insomnia, Gerstmann-Strasser-Schlegel disease, Creutzfeldt-Jakob disease, where Creutzfeldt-Jakob is important, we usually asked for the exams. Then in animals, infectious prions cause mad cow disease. You might have heard about mad cow disease. Mad cow disease, then scrapie in sheep and goats, and chronic wasting disease in elk and deer. And TSCs can be transmitted between animals and from animals to humans by eating contaminated meat or animal feed. So it is transmittable. Like all other viral diseases, this disease is also uh, transmittable from animals to humans. And there is no evidence for transmission via casual contact with an infected person. And this is the picture showing a normal brain and a brain infected with uh, Creutzfeldt-Jakob disease. That is, the brain tissues become much softer. Certain holes you can see here, more holes are developed. So the function of normal brain gets altered. Creutzfeldt-Jakob disease is a fatal disease that causes the degeneration of a neural tissue. These brain scans compare to normal brain with one with CJD. CJD means Creutzfeldt-Jakob disease. And compared to normal brain, the brain tissue of a CJD patient is full of sponge-like lesions, which results from abnormal formation of prion protein. So hope you have understood what happens when infectious prions infect your brain. The normal brain become spongy, and numerous lesions develop in the brain tissue and it become functionless. Then you can study in detail about Creutzfeldt-Jakob disease, or it is also known as bovine spongiform encephalopathy in humans. Okay, how this Creutzfeldt-Jakob disease is transmitted into humans? Because mostly through eating diseased animal food. For example, by eating infected beef, you can get this disease. That is by eating infected beef having a blood transfusion, or from a person with a disease, or injection of human growth hormone extracted from the bodies with the disease, or through infected surgical instruments. All these are the ways in which this disease gets transmitted into human beings. Patients suffer from ataxia or disequilibrium. What do you mean by ataxia? Ataxia means a patient cannot stand or walk well because he cannot maintain his equilibrium. This is because of a disease in cerebellum. Okay, so causes ataxia and dementia. What is dementia? Dementia is loss of mentality or progressive loss of cognitive function and usually die after 1 year. Okay, that is the brain function gets completely altered and the person become unable to speak or walk and he completely lose all his memories. That is the condition known as dementia. And ultimately he will die soon. That is within few months the person will die. So these are the human prion diseases. And the most dominant disease is Creutzfeldt-Jakob disease. Then Kuru disease. Kuru disease also you might have heard about Kuru disease in your plus two classes. I think you might have studied Kuru disease. That is, this disease is caused when we eat the brains of dead people. And that is uh, why it is not a it is not a normal practice to eat the brain of dead people, isn't it? But in certain places of Africa or in New Guinea, it is a practice as a part of their funeral rites. It is a practice of eating in the brain of dead people. So what happens when when they eat the brain of dead people, this disease get transmitted into their body. And what happens when we so what is the after effect? After effects are ataxia and dementia and the ability in moving their eyes. And the person or the patient will die within 2 years. Then another human prion disease is Graceman-Schlocker-Schringert syndrome, and is a very rare syndrome. And the after effects are ataxia and dementia. Like so, the Creutzfeldt-Jakob disease, Kuru disease, then Jaceman-Stosser, Schlinger syndrome. For all these diseases, the after effects are ataxia and dementia. And gradually the sudden death is also the cause of this disease. So that is, the patient will die within 6 months or within 1 year. Then, what are the diseases caused to animals due to infectious prions? That is, first one is bovine spongiform encephalopathy, then feline spongiform encephalopathy, ungulate spongiform encephalopathy, then chronic wasting disease or CWD. And bovine spongiform encephalopathy is commonly known as mad cow disease. And mad cow disease is a disease found in cattle. It is a progressive neurological disorder of cattle that results from infection by an unusual transmittable agent called a prion. So it is a neurological disorder found in cattle. And how this disease is get transmitted into cattle, or how cattle get this disease? That is, it is due to the feeding of certain protein supplements contaminated by materials from infected animals. We feed certain protein supplements to the cattle. So in that feed, if that feed is contaminated with the diseased animals particles or some materials from the diseased animals, then usually the cattle gets this disease, and that disease is known as mad cow disease. It is a very severe disease found in cattle. So bovine spongiform encephalopathy or mad cow disease. It is a disease due to prions in animals. Most important disease found in animals due to prions. So hope you have got an idea about prions. Okay. So in prions is important, and you can expect a short answer question from this part. That is, what are prions, and the name of the scientist who discovered prion, and What are the diseases caused by prions? What are the human diseases caused by prions? What are the diseases caused to animals due to prions? All these are important. You can expect for this for your for some university exam for the in the microbiology paper you can surely expect a question about prions. Then next is viroids. Next subviral particle we are going to study is viroids. And the viroids uh, was first discovered by Theodor Dina in 1971. And he is a pathologist working at the Agriculture Research Service. And he discovered an acellular particle and he named it as viroid. And viroid, the word meaning of viroid is virus-like. Word meaning of viroid is virus-like, and it was discovered by Theodor Dina. And it consists of uh, viroids. It consists only a single-stranded RNA or a short strand of circular RNA capable of self-replication, no double strand or no DNA. It consists of only a short strand of circular RNA which is capable of self-replication. This RNA is capable of self-replication. Very, very important. Okay, because after viroids, we are going to study another subviral particle that is virusoids. So when we study that virusoids, you need to compare between viroids and virusoids. So don't get confused. This viroids, it consists of only a single RNA, single-stranded RNA, which is capable of self-replication. And the first viroid discovered that was found to cause potato tuber spindle disease, which causes slower sprouting and various deformities in potato plants. So the first viroid discovered was known to cause potato tuber spindle disease, and the potatoes infected with this viroid disease uh, show slower sprouting. They sprout very slowly, and various another many other deformities were also found in the viroid affected potato plants. And same like prions, viroids are also very, very smaller than viruses. So the exact definition of viroids is they are small single-stranded, very important, small single-stranded circular RNAs. It is not DNA. Okay, it is a small single-stranded circular RNAs. Example, the length is approximately about 50 nanometer, and that are infectious pathogens. So it is a small single-stranded circular RNA about 50 nanometer in length, and there is no coating. Viruses have a protein coating, but viroids, they does not contain any protein coating. So that is also important. So viroids, they contain only a single stranded circular RNA capable of self-replication, and they do not contain protein coating. The RNA is not enclosed in any structure and except during infection is not associated with any other chemical substances. So there is no protein coating in viroids. And mostly viroids, they cause large losses to agricultural crops, especially potatoes. They also infect the cucumbers, tomatoes, chrysanthemum, avocado, coconut, etc., palms, etc. And the picture shows potatoes infected by a viroid. So how this potato spindle tuber viroid is spread? Usually, it is usually spread by using infected knives. We usually use knives for cutting the matured potatoes, isn't it? So when we use infected knives for cutting the healthy potatoes, then the disease get transmitted into potatoes. So just remember the other plants gets infected with the viroids, that is uh, potatoes, then cucumbers, tomatoes, chrysanthemum, avocado, coconut, palms. Then viroids can result in devastating losses of commercially important agricultural food crops grown in fields and orchards. Since the discovery of potato spindle tuber viroid, other viroids have been discovered that cause disease in plants. Just uh, go through the names. It is not much important, but just for your information, just go through the names. That is tomato planta macho viroid, which infects tomato plants. And what are the losses caused due to this infection? What are the losses caused due to tomato planta macho viroid infection? It causes loss of chlorophyll. Chlorophyll gets lost. Then the fruit become disfigured and leaves become brittle, and the size of the potatoes also gets reduced. Then avocado, avocado sunblosh viroid. So uh, mostly it lowers the yield and lowers the quality of the fruit. Then peach latent mosaic viroid. It causes necrosis of the flower buds and branches and wounding of the ripened fruit, and that infection also causes certain fungal and bacterial growth in the fruit. So we can say viroids in general can be dispersed mechanically during crop maintenance or harvesting, vegetative reproduction, and possibly via seeds and insects, resulting in severe drop in fruit availability and devastating economic consequences. So there are different ways in which viroids can be spread. That can be due to by means of vegetative reproduction, or through seeds or insects, etc., or the through infected knives used for cutting fruits, etc. Okay, so viroids. Only for prions, periods, etc. Only short answer questions will be asked. That is, write short notes on prions, periods, virusoids, etc. Only such questions will be asked. Okay, so you need to study only that much. In your syllabus also, very brief study is mentioned. So the next subviral particle is virusoids. So don't get confused with viroids and virusoids. Okay, they spell similar, so do not get confused with their functions and their roles. So we can study by comparing between viroids and virusoids. We said viroids are self-replicating, but virusoids are not self-replicating. Okay, and the similarity is that both are single-stranded RNAs. And in viroids, there is no protein coating. But in virusoids, they are encapsulated by a helper virus coat protein. So, virusoids, they have a helper virus coating. So, what are the differences between viroids and virusoids? Viroids, they are self-replicating. Virusoids are non-self-replicating. And viroids, they do not contain any protein coat, but virusoids, they are encapsulated by a helper virus coat protein. And what is the similarity? Both are single-stranded RNAs. So since virusoids, they are non-self-replicating, they depend on, so how they can replicate, they depend on this helper virus coat protein. So that is how they replicate. Since they are non-self-replicating, for the replication purpose, they depend on their helper virus code protein, and they can infect commercially important agricultural crops. The genome of virusoids, they consist of several hundred nucleotides. For example, about 200 to 400 nucleotides, and they does not code for any protein. And the genome serves only to replicate virusoid RNA. Both virusoids and viroids encode a hammerhead ribosome. That about hammerhead ribosome and all you will study in detail in your second sem molecular biology portion. Okay, so no need of going into detail here. And the first virusoid was discovered in Nicotiana plant. In Nicotiana verutina plant, infected with velvet tobacco mottle virus R2. So the first virusoid discovered was in Nicotiana velutina plant. And now only 5 types of viruses were discovered, and their associated helper viruses. And all the helper viruses associated with viruses, they belong to the family of the viruses. And you can also study some examples of helper viruses. For example, subterranean clover mottle virus, which has an associated virusoid packaged inside the viral capsid. So how this helper virus coating, how it helps the virus out to replicate? That is, once the helper virus enters the host cell, the virusoids are released, and they can be found free in plant cell cytoplasm. Then the remaining activity, that is the remaining replication activity, is performed by the helper virus coating. So, since this virusoids, they do not have the capacity of self-replication, the process of replication is performed by their helper virus coating. And almost all the helper viruses associated with virusoids, they belong to the family of viruses. For example, an example, it is subterranean clover mottle viruses, and which is the first virusoid discovered was in Nicotiana plant. And we have already said that viruses depend on the helper viruses for their replication. So, because of this property, they are classified as satellites. So, they belong to a large group of infectious agents called satellite RNAs, which are similar to pathogenic RNAs found in animals. Then, an example of a satellite RNA that infects humans is hepatitis delta virus or hepatitis delta virusoid. And much larger than a plant virusoid, hepatitis delta virus has a circular single-stranded RNA genome of 1007. And the HDV helper virus, another one is hepatitis B virus. And so an example of one satellite RNA that infects humans is hepatitis delta virus or hepatitis delta virusoid. And hepatitis delta helper virus, example of a helper virus is hepatitis B virus. So when this hepatitis delta virus and hepatitis B virus, uh, it co-infects an organism or a plant, the infection is very harmful. So, for virusoids, you need to study only what is its definition and what are the differences between viroids and virusoids, and what is the structure. Structure means how many nucleotides were found in virusoids, and what is the role of helper virus coating in virusoids, and what are the study one example of helper virus or belongs to the family of Subviral. You can study one example, that is subterranean clover motile virus, etc. So that is all about the subviral particles, prions, viroids, and virusoids. So you can expect a short answer question, or write a short note on. You can expect a question like this: write a short note on prions, viroids, and virusoids. Okay, so study uh, what I have given in this slides is enough. Is much and enough for your exam. Okay, so it's a very important topic. So study well. Thank you.